laboratory supervisor telling me that if there's one thing I should never do, it's stand behind a podium. People my height can't get away with it very easily. But I'll give it a go today. It's a great pleasure to be here with you all for World MS Day. I am a neuroscientist and a stem cell researcher, and I put those specialisation, areas of research in the direction of studying MS. And what I want to talk to you a little bit about today isn't just my research, but the research that's been carried out into MS across the Menzies Institute. And part of that has involved me being pushed a little bit, we talked a little bit earlier about silos, being pushed a bit outside of my comfort zone, particularly in the last 12 months, as we're coming together to try and work across the different disciplines to coordinate MS research, to try and move more things through, to translate that research to actually have impact and benefits to improve the lives of people with MS. And in doing that, I'm working with, there we go, a number of people in the Institute, and this is a photo that was taken on Red Lab Coat Day for MS Research Australia. And it shows a number of the people working in the Menzies Institute on MS research. I'm here in the front, and as I said, I'm a laboratory researcher, so most of the work I do is trying to understand how cellular changes can actually result in MS. But I'm now working very closely with Ingrid van der Meij, whose research primarily focuses on understanding how lifestyle factors can either increase a person's risk of developing MS or even change, the progression or course of their MS. Along with Andrew Palmer, who is a health economist. He provides very useful information about how much multiple sclerosis costs individuals, costs per individual for the community, so that we can actually try and explain to government and other organisations why it's really important to help support research into MS, that actually it, and also to help us justify why, when we develop a new treatment, that treatment is actually going to be beneficial if the government supports that treatment so that people can actually obtain it. And we also work very closely with Bruce Taylor and Jack Charlesworth, who are shown in this image here. Bruce Taylor is a neurologist who works at the Menzies and also at the Royal Hobart Hospital. And many of you probably know Bruce. He has been involved in MS research in Tasmania for quite a long time now. And he is a clinical researcher and also a genetics researcher, so he tries to understand how genes impact the development of MS. And Jack Charlesworth works with him very closely, actually, in trying to understand how genes result in the development of multiple sclerosis. So, I'm actually going to have to move this a little bit. So, in coming together, we've actually developed what we're now calling the MS Research and Translation Network. There was a little bit of debate as to what we should call this, and I want to explain to you today a bit about what that actually is. So, in talking about how we can improve MS research and also the use of that knowledge that we gain through research to actually make it beneficial for treatment, our mission was to generate and translate and apply the knowledge so on the cause, treatment and prevention of MS, so that we can actually meet our vision, which is to reduce the impact of MS for individuals and the community. And within the MS Research and Translation Network, our focus is across a number of different areas. So we're trying to understand the biology of multiple sclerosis. A big part of that is trying to go to this question of what is the cause of the disease, which is something that after many years of research we still don't know. We're also trying to identify things that could be modified through we identify genes in our genetic studies, and we're trying to find out how we can use that information to actually develop treatments, modify those signalling pathways to develop new treatments. How can we prevent disease? So if we can understand more of the factors that contribute to the risk, can we then actually use that to promote prevention? And also we want to try and decrease the societal impact of the disease overall by applying this knowledge. Okay. Sorry, I'm having a little trouble because I can't see the screen there versus what I have here, and I'm really sorry about that. I will try and do better. Okay, so we're hoping to actually launch the MS Research and Translation Network later in 2018, and at the moment it is The cluster of research is at the Menzies, but throughout 2018 we are now inviting other organisations across Australia, particularly researchers in Perth, Canberra, Sydney, Melbourne, to join in this research network. And then once we actually get the national team built, we're looking at expanding this overseas. And these arrows are not specifically indicative of countries, just the fact that actually we're trying to build an international network. And in terms of who we're asking to join the network, it's not individual researchers. Researchers will absolutely be involved in the projects on an international scale, but what we're trying to do is actually get buy-in from the various universities, hospitals, charity organisations, and including MS Research Australia, MS Limited, to be part of this network so that we can actually try and take a more holistic approach to MS research in Australia and overseas. So, what are we doing to achieve the vision? It's all very nice to say we're going to try and get lots of people to come together to work on research, but unless we actually have a very clear plan for how we're going to make that work, it may not actually amount to very much. And so we've been looking at developing our strategy. First of all, we're establishing a world-first MS stem cell repository. It's one of the things that we're doing. I just want to give you a couple of examples today. And we're calling this MS STEM, and I'm going to talk to you a bit more about that. Another thing that we're doing is developing an educational online course. It's going to be called Understanding MS, that will actually be an educational and a research tool. And the third thing that we're doing is we're going to be actively translating MS research findings to ensure they benefit people with MS. And one of the first things that we're taking through this translational pathway is a clinical trial of transcranial magnetic stimulation, and I will elaborate on that a little bit too. So the first thing, as I mentioned, is an MS stem cell repository, what I'm calling MS STEM. And this actually involves, creating one of these stem cell banks, involves taking skin biopsies from people, taking the cells out of these skin biopsies and growing them as what we call a fibroblast cell line, then reprogramming these cells. So fibroblasts or skin cells, they're mature cells, and we're actually going to reprogram them to make them into stem cells. It is now possible to take fully mature cells in the body and make them, go backwards through maturation to become a stem cell again. And in doing this, we can create a series of stem cell lines from people with MS. And we've already started doing this for some people, and we've been collecting some of these stem cells. But actually what we're aiming to do now is to upscale this so that we can generate and store stem cell lines from 100 people with multiple sclerosis, 40 from people with primary progressive MS, and 60 from people with relapsing remitting MS, and then 40 lines from people that don't have any neurological condition. Now, this is actually quite a large undertaking, and so if the process that I've initially described, taking skin cells and making them into stem cells, it's actually quite involved and can be quite time-consuming. But we're developing a new process. Researchers have recently published it's possible to take blood cells and directly make those blood cells into stem cells. And if we could do that, it's much easier to ask people to provide a blood sample for us to then create these stem cell lines. And we're hoping to then be able to do this on the large scale that we're trying to achieve. And the other advantage of this is that we can then use these stem cells to make brain cells. One thing about MS is that you can study immune cells relatively easily. You can take immune cells from the blood. But a lot of the problem we have in studying with MS is that it's really hard to study what happens in the brain. You can image the brain, but that doesn't really tell us how the cells are changing. Not on an individual cell level or on the level of genes or proteins. And so if we can take these stem cells and actually make them into the cell types of the brain that are relevant to multiple sclerosis, we can study them directly. And one of the cell types that is very relevant is the oligodendrocytes. They're the cells that provide the insulation in the brain, and they die in multiple sclerosis, and they cause the insulation to be stripped off of the nerve cells. And one of the things that we can now do is take these stem cells and mature them into oligodendrocytes. This wasn't possible until relatively recently. We have collaborators in Germany that have been training my postdoc, Kimberly Pittman, who's in the red shirt in this photo, how to actually make the stem cells into oligodendrocytes so that we can look at these cells in a dish a lot more closely. And so this is where we're going. But I want to tell you guys a bit about what the value of having this stem cell bank would be. One of them, as I've already said, is that we can actually study the brain cells themselves, which allows us to look at a lot of different things that we couldn't look at before. So we can look at the neurons and the oligodendrocytes, and we can see how their health, their behaviour, the genes that they express can all change, and in doing that, hopefully try and also get at some of the cause of MS. So why is it when the immune cells get activated that they actually go into the brain and attack the brain in the first place? We hope to also be able to study the effect of the MS-associated genes. So one of the things that we can say at the moment is that there are a lot of potential genetic variants that could contribute to MS, but actually proving that they are a cause has been extremely challenging. One thing that we can do now is use a technology called CRISPR to actually correct that mutation in the DNA of a cell and then see, does that fix the cell? Does it make it behave normally again? And if that's the case, that says that that gene really, really was involved in causing the phenotype or the change in MS. So the other thing we use them for is that a source of human cells for testing new drugs for MS, testing new treatments, is really important. We can use mouse cells for some things, we can use cell lines for other things, but human cells sometimes behave a little bit differently, and having those cells that we can study directly will be really valuable. And we also hope to use them to try and get at some of the distinct differences between primary progressive and relapsing remitting MS. So, how am I going for time? I'm good? Okay. So one of the examples, I guess, that we're starting with in terms of the first uses of the stem cell bank is actually to try and look at identifying specifically which genes might be really important for developing or causing MS. We know that MS risk is about 1 in 750 for the general population, but it increases to 1 in 40 for if you have close family members with MS, or 1 in 5 if you have an identical twin. So these statistics, these numbers, they tell us that there is some genetic component to multiple sclerosis. And so we really want to know which genes are really behind all of that. And we can do this, we've been starting these studies by doing whole genome sequencing, and various people in Tasmania have given blood samples previously for us to carry out some genetic studies. And by performing sequencing of like the entire DNA so that we know all of the coding sequence, we can actually look at particularly family members where there is a high incidence of MS and try and identify which changes in the DNA might be responsible. And one variant that we've identified that we want to try and test, so at the moment this is something that may or may not cause MS. When we've run the statistics it looks pretty likely, but until we can do the experiment to test it directly we don't really know. And this is where the stem cells come in. So one of the variants that we've found, it's the gene that's called GRIC4, and there's one particular part of the sequence that is changed, and it suggests that that sequence might then change how this gene functions. But it doesn't seem to be a gene that's expressed in the immune cells. It seems to be a gene that's highly expressed in the oligodendrocytes that I mentioned earlier, which are the cells there with at the bottom of the diagram. I can't really point from here. Or can I point? Oh, I can point. Does that come up on there? No. Okay. And the other cell type that expresses this gene really highly are the OPCs, which were mentioned in my introduction. They're kind of like a stem cell that we have in our normal brain, and they're the cells, like normal, healthy, all the time, they will divide and they will make new oligodendrocytes. Now, in multiple sclerosis, they are having to work overtime because they have to make new oligodendrocytes to try and replace the ones that are lost. And this process doesn't always work particularly well. And so it may also be that a failure of that repair process can also contribute to disease. And so we're trying to understand how GRIC4 could be affecting these cells, and we're using stem cells to do it. Now, GRIC4 is a gene that results in a protein on the cell surface, and it can regulate how excitable cells are in the brain. So whether or not they can respond properly to the chemicals that the brain uses to communicate between cells. And so we're trying to study this, and we're studying this now using these stem cells. So we've generated stem cells from individuals with MS that carry this mutation, and we're using them to make the brain cells. And we can also study cells with the mutation to see if they behave differently to the brain cells from people without MS. And we can do this directly by using this technology I mentioned called CRISPR. So if you look here and you can see in the first example, there's a little T that's in yellow. If this is a DNA sequence, that could be the one change that we have identified. And now in a cell line, we've got the ability to go in and use an enzyme to basically correct this in the cell. So we make that T that is the wrong thing go back to being the G that is the right thing. We can also take cells that have the normal sequence and introduce that single mutation, so that then we can find out if putting that one change into the DNA is enough to actually produce multiple sclerosis. And we can try and model this in culture. So another thing that we're producing through the MS Research and Translation Centre is this a massive open online course called Understanding MS. So this has been in collaboration with MS Limited, and it's going to be available to anyone with an internet connection. And it has, it'll be a 6-week course that has a number of aims, and it'll basically take people step by step through and provide information about multiple sclerosis. So in some ways it's an educational tool. And we aim to create this educational platform so that we can increase the level of accurate and current information that's out there about MS. If you Google something about multiple sclerosis, you can get a whole bunch of different hits. What we're trying to do is provide a way that people can get information that has been scientifically tested and validated, so that we know that actually what they're looking at is the current of the field as well. So it's basically where researchers say, I understand we're at. And we want to also be able to use that then as a way of also collecting additional research information. But the audience will be people living with MS, their families, GPs, allied health professionals, and advocates and other local area coordinators. And we're hoping to have the pilot for this MS MOOC out by October this year. And the target date for the live public launch is the 1st of March in 2019. So keep an eye out for it. Okay, so I have one last thing that I wanted to cover briefly, and that is the clinical trial that we're starting as part of the Translation Centre. So as I mentioned, in MS, oligodendrocytes can die, and oligodendrocyte progenitor cells are stem cells that we have in our own brain that naturally try and repair it. One thing that we know about these cells is that they respond really well to activity. So if the nerve cells have a particular pattern of activity, the stem cells recognise that, and it tells them to go on and mature and make new oligodendrocytes. And we wanted to try and harvest this knowledge and apply it so that we could actually see whether or not we could increase the chances of these OPCs making new oligodendrocytes. One of the ways we thought we could do this is to, I guess, almost massage the activity of the brain and apply a healthy pattern of activity, and we're doing that using TMS, transcranial magnetic stimulation. It's basically a magnet that is put over the head. You don't really feel anything. It's such, we actually have it at a very, very low intensity, but we found that it's actually enough in preclinical trials to double the number of new oligodendrocytes that get made inside 2 weeks. And in preclinical trials, we've also shown that within 10 days, it can about halve the size of lesions compared to the animals that had just sham stimulation where the magnet's over the head but not turned on. And because this has been used previously in the clinic for the treatment of depression, I'm working very closely with Bruce Taylor and other collaborators to actually try and take this through to carry out a safety trial later this year for people with MS. We also want to use this as an opportunity to try and collect early measures of efficacy, those early indicators as to how well it might be working, so that we could then have the data we would need to try and move this forward. And it basically involves recruitment of people through the Royal Hobart Hospital. We will be carrying out an initial MRI to get a scan to see where the lesions are and what size they are at the beginning. We'll ask people to actually undergo an initial fatigue assessment as well. We will then apply the TMS to people for 2 weeks or 4 weeks. And then repeat the MRI so that we can see if the brain lesions have gotten smaller or not changed, or also assess people's fatigue and see whether or not that has changed at all during the course of the test. What we're basically looking for is any early signs that this particular treatment can do what our preclinical tests say, which is try and push the repair of the brain lesions. And so we'll be commencing this early trial in October of this year. And that's all I have to say. Thank you.
Stay up there, Kaylene. So we don't have a lot of time for questions, but I think there are lots of information. So one of the key questions would be, how can people get more information?
So if people want more information about any of the things I spoke about, they can email myself or they could email Bruce Taylor. And we will certainly be providing more information through the Royal Hobart Hospital Clinic as well as part of some of the participant recruitment for some of the studies. So for people to donate for the stem cell bank or participate in the TMS trial, that information will be available through the MS clinic. And when it comes to the MS MOOC, we will be advertising that through the MS societies, through MS Research Australia, MS Limited, and also hopefully some media in Tasmania in leading up to that, so that you will all be made aware of the fact that, I guess, of how the pilot went and then the rollout of the actual full MOOC.
Fantastic work, and we really appreciate all your work, because without you, we would be struggling for both interventions and future prevention. So thanks, Kaylene.